Welcome back, students. Welcome to Priya's Lifescience Academy. This is your educator Priya. Today, I'll teach you another very important operon, which is known as TRP operon or tryptophan operon. Okay. Unlike lac operon, we have seen that in case of lac operon, it was an inducible operon. But this operon, the TRP operon, is a repressible operon. Okay. It's a repressible operon because the operon needs to shut off. We have seen that in case of lac operon, in initial stage, lac operon was turned off. It needed to turn on by the help of the effector molecule, that is lactose. That is why it was known as inducible operon. But in case of this TRP operon, this operon needs to turn off. That is why it is known as repressible operon. Initially it is turned on, but it needs to turn off by help of another effector molecule, that is the tryptophan itself. So that is why it is known as repressible operon. Now, coming to the main structural part of the operon, you know that operon contains many genes together, and it is under the control of a single promoter operator region. In case of TRP operon, you can see TRPA, TRPB, TRPC, TRPD, TRPE. 5 structural genes are under the control of a single promoter operator region. Okay, so this is the concept of the operon. I hope you remember now. And from these, from this operon, you can get polycistronic mRNA, and then from polycistronic mRNA, you can get the proteins. Okay. So from TRP operon, the enzymes are produced. Those are known as anthranilate synthase 1, anthranilate synthase 2, RP isomerase NGP, tryptophan synthase beta, tryptophan synthase alpha. These enzymes work in a pathway, and their end product is tryptophan. So the name suggests the tryptophan operon, the end product of the tryptophan operon works for the production of the amino acid tryptophan. We know that tryptophan is a very important amino acid. It is needed in protein production. So the end product of this tryptophan operon is tryptophan. Okay, and in the operon, there is another structure that is known as leader region. Between the operator and the structural gene, there is a region known as the leader region. This region is very much important for the regulation of TRP operon. So this is the basic of TRP operon. You have 5 structural genes. Those are under the control of a single promoter operator region, and these structural genes are needed for the production of the enzymes. Those enzymes will work together in a pathway for the production of the tryptophan amino acid. Okay, so this is the overall picture of the tryptophan operon. Now, coming to the regulation of the tryptophan operon. We have studied about the regulation of the lac operon. Now, we have to study the regulation of the tryptophan operon. At initial stage, tryptophan operon is turned on, you can see. But when there is an ample amount of tryptophan is present in enough amount, the operon needs to turn off. That is why it is known as repressible operon. And to turn off the tryptophan operon, it needs the tryptophan itself. Okay, that is why this operon is also known as end product inhibition, because the end product of this tryptophan operon is tryptophan, and the end product is helping the operon to turn off. That is why it is also known as end product inhibition. I'm giving you a very easy example for this, like your mother has prepared kachori and sabzi for dinner. So she has given you 5 kachori, but after eating 4, you are feeling full. Your stomach is full. You can no longer eat. So the 5th kachori, you are returning it to your mother. Mama, I'm full. Please don't give me any more. Same is the case in case of tryptophan operon. The end product tryptophan, when it is present in high amount, it is acting as a feedback to the operon to turn it off. So it is binding on the operator region to block the binding of the RNA polymerase on the promoter region, like in the lac operon we have seen that once the lac repressor is bound on the operator, the RNA polymerase cannot bind on the promoter region and transcription is halted. So as the case in case of tryptophan operon, the end product is giving a signal that enough tryptophan is present, no more production of tryptophan is needed, so it is acting as a repressor and binding on the promoter region to block the transcription. Along with tryptophan, there is another repressor that is helping the tryptophan. So in the upstream of the tryptophan operon, there is a sequence. That sequence is responsible for the production of the Appo repressor. Okay, it's present. It's present a long distance away. That is why it is not drawn in this picture. But there is one region present upstream of this operon that is responsible for the production of the Appo repressor. Okay, but the Appo repressor is inactive. It can't bind on the operator and turn off the transcription. But whenever tryptophan is there, Appo repressor plus tryptophan, it is producing active repressor. So the thing is that Appo repressor is always present, but it can't bind on the operator region and can't turn off the operon. Whenever Appo repressor is conjugated with tryptophan, it is producing the active repressor. Then the active repressor can bind on the operator region and can turn off the operon. Okay, I am giving you another very clear description. That is, these you know in case of lac operon as well. This is promoter, this is operator, and this is the leader, and these are the 5 structural gene. 1, 2, 3, 4, 5. Okay, this is the 5 structural gene. So whenever Appo repressor binds with the tryptophan, they both form a complex and they bind on the operator region. So the operator is blocked now. RNA polymerase can't bind on the promoter region and transcription is halted. So by this method, tryptophan is helping the Appo repressor to turn off the tryptophan operon. I think you have understood now what is end product inhibition, because the tryptophan is the end product and the end product is helping to turn off the operon. So this is one regulation of the tryptophan. So I am giving you another very quick revision, that is, in initial case tryptophan operon is turned on, but when enough tryptophan is present, tryptophan binds with Appo repressor and it binds with the operator region to turn off the tryptophan operon. Okay, then the enzymes are not made, and if the enzymes are not made, tryptophan will not be produced. So this is one regulation of the tryptophan operon. There is another regulation of the tryptophan operon by this leader region and by this attenuator also. Okay. So I'll come to the next class. So for this class you have understood the regulation of the what is tryptophan operon, what is the regulation of the tryptophan operon by operator repressor. So first regulation is by operator repressor and the next regulation is by the leader region. I'll come to this in the next class. I hope you understood, students. Thank you for watching. Keep learning, and as I always say, fight for your dreams will fight for you. Thank you, students.